would like to introduce our first speaker, Dr. Rachel Idle. She is an assistant research scientist in fruit production and genetics at the University of Georgia.
Thank you. Well, good afternoon, everyone. Today I'd like to talk a little bit about some blueberry fruit quality work that we've been doing in my lab, an update on the flavor profiles for consumer preferences, and some preliminary data on the maintenance of these flavor profiles over post-harvest storage. So first of all, just a general overview. What is fruit quality? It is basically a complex trait, and it can be considered whatever the consumer desires. And the attributes that basically contribute the most to fruit quality, the number one is appearance. You look at a fruit before you eat it, appearance and color, texture, flavor, which we're going to go into today, and nutritional value. Oops. So one of the biggest things that contributes to the overall liking or how much a consumer likes the blueberry fruit is sweetness and flavor. So what is flavor? It's composed of two attributes, taste and aroma. And taste can be influenced by or controlled by sugar content and acid content, and aroma, the volatile concentration or what we smell. So basically, research really needs to focus on both non-volatile, sugar and acids, and volatile, what we smell, to kind of get a holistic idea of what's going on with the aroma of fruit. So again, the flavor is a mixture of these two items. So taste is what we perceive in our mouth, and aroma is what we perceive through our nostrils. The aroma is derived from what's called volatile compounds. So these are chemical compounds that make up flavor. And some fruit can be based on, the aroma can be based largely on one compound, such as banana or peach. And some fruits can be multiple compounds contribute to the aroma, like citrus, apple, or tomato. So for those of you who may not be aware or may not be familiar with blueberries, because I know we have a lot of sessions here, basically, why do we care about blueberry flavor? Why are we looking at this? Well, to take a step back, there are three major commercial blueberry types grown in the U.S. and really worldwide. Within Georgia, we grow two of those types, southern highbush and rabbit-eye, and there's a third commercial type, the northern highbush type. There's often an idea that exists, that it's a subjective idea, really, that northern highbush fruit have higher quality than southern highbush and rabbit-eye types. This is largely unfounded, as there's not a lot of work that really supports any of this, or cultivars are outdated and so forth. And the reason why this is important, the main take-home, is it may lead to lower price point received from our growers for southern highbush and rabbit-eye, or complete cultivar exclusion for no real reason other than a subjective bias. And it's often thought that northern highbush are just overall superior, and rabbit-eye, for example, are inferior, and southern highbush is kind of somewhere in the middle. So today I'm going to talk mainly about the relationship between flavor compounds and consumer acceptability, as we just recently finished up some of that work with one of my PhD students, and give you some preliminary data on the flavor volatile changes over cold storage as we're working to finish that project. So we collected this fruit in 2018 and 2019. We've done a number of projects that I've talked about over the years here with that fruit. This is one of those projects. We took that fruit back to campus, and we did two sections. I forgot to put that in this slide, but one was kept fresh for the consumer acceptability, and then the other was put in the freezer until we processed it for the chemical attributes. The main goal that we sought to do for cultivars was trying to get a nice representation of the current market. Of course, these are not an exhaustive list of all of the cultivars, but some of the major cultivars. And the ones in purple were in common across both years. So we had 13 cultivars in common across both years for the three commercial blueberry types. So the first and the main part of this presentation, looking at flavor components and acceptability scores, those are two of my students at the top, Jamal and Kathleen, who did the work. We started with consumer sensory studies. So again, this was fresh fruit. Fruit were washed and sorted, always making sure to give each cultivar a good fighting chance. So the best representation of that cultivar no underripe or overripe fruit, so just really pretty berries for each cultivar. We brought it to room temperature, and we did these panels over 2 years. We invited the same panelists, so it was the same panelists over year 1 and year 2, and across multiple sessions, because as you know, the cultivars and the types are not all ripe at the same time. Plus, it would be too much to ask someone to evaluate all at one time. We used what's called a hedonic scale. I can't really see my mouse, but it's there at the bottom right underneath the pictures. It's a score of 1 to 9, with 1 dislike extremely, 5 neither dislike or like, and 9 like extremely. And then to process, to understand a little bit more about the chemicals involved, we extracted the flavor. I don't have a pointer, so I can't circle things. But basically, we went ahead and took the fruit that was frozen, we thawed it, blended it up into a puree. For the flavor volatiles, just like if you're making, if you're making cookies over the holidays, when you put them in the oven, the heat, you smell the chocolate chip cookies everywhere. So we put them in the little shaker, basically heated it up and shook it so we could get all of those wonderful smells out of the blueberries. Put a little fiber in that container and then put it into the machine, and in H you can see the individual compounds coming out to show the individual flavor volatiles. We also took a look at the overall estimates for titratable acidity, so fruit acid content, and soluble solid content, also known as Brooks, which is basically overall estimate of sugar content as well. And then lastly, we extracted, just like we did the flavor volatiles, we wanted to get each individual compound. We did that for sugar content, so sucrose, glucose, and fructose, and then 5 organic acids as well. We took out little samples of blueberries and put them in the machine and separated out individual compounds for those. So on to the results. So this is, these little plots are called spider plots. Or it can be called spider plot, but the basic idea is you see the pentagon there. The outside, the closer it is to the edges, the higher the scores. The closer it is to the inside, the lower the scores. So when we looked at the types first, so Rabbit, Southern Highbush, and Northern Highbush cultivars, and again, I don't have a pointer, so I apologize about that, but Rabbit Powder Blue had the highest acceptability for overall liking and for flavor liking. Southern Highbush, it was Legacy and Camellia, and Liberty for Northern Highbush. When we put all of these together for all of the types, Legacy, Camellia, we had Legacy from Michigan and also from Georgia, but Legacy and Camellia, the 2 Southern Highbush, come out on top. Then we looked at it by type, and our results suggest that Southern Highbush ranked higher for overall liking than Northern Highbush and Rabbit, and also for flavor liking, Southern Highbush ranked the highest. Rabbit ranked the highest, as you can see that blue going up to the top there, for aroma liking. So results that we've obtained, it contradicts the subjective perception that Northern Highbush have better fruit quality, especially for flavor and overall liking, than Southern Highbush and Rabbit. Then we went ahead and took a look at how of these scores that the consumers are providing us, how do they relate to one another? So we looked at aroma, the how much, and each one of these is how much do you like it, right? 1 to 9 scale. So aroma, flavor, sweetness, acidity, and overall liking. And we found that overall liking was strongly related to flavor, sweetness, and acidity. Sweetness was strongly correlated or related to flavor. And flavor might be the most influential category in human preference for fresh blueberries. And our results suggest that these taste-related traits, sugar and acid content, may highly influence flavor acceptability, not just the flavor. So these are just 2 kind of busy pie graphs. The main take-home for this is we put these samples in the machine and we looked at all the results that came out, and we looked at all the different chemical groups that contribute to the aroma, the smell of blueberries. We found over 65 volatile compounds, so 65 compounds coming out of the blueberry samples contributing to aroma, and they were sorted into 8 major chemical groups. Out of those 8 groups for both years, 2 groups, terpenoids and aldehydes, you don't have to remember these names, but 2 groups came out on top, the terpenoids and the aldehydes. These 2 groups, chemical compounds in these 2 groups, accounted for over half to over 80%, so 60 to 80% of all of the volatile compounds, all of the smell compounds coming out of blueberries. And these compounds, chemicals in these groups, give the sweet, the floral, the fruity notes, and in the aldehydes are the green or the grassy notes. So these are the main compounds that we should be looking at when examining the flavor. Okay, so in this, the main idea is this is a cluster, it's busy, but I'll give you the take-homes from it. The majority were these terpenoids, so basically this one group that gives you the sweet, the floral, the fruity. These compounds were found to be related or correlated to the aroma attributes, so how much they liked overall liking and so forth. The interaction of these volatile compounds may influence aroma and flavor of the fruit. No other relationships between the other volatile compounds were established, so those smaller groups, we also looked at those. Okay, then we took a look at how the overall sugar content and overall acid content broke down across the types. Soluble solid content was variable in blueberry types across the years. Titratable acid content was the highest in northern highbush as compared to the other types for both years. Sugar and acid ratio was the highest in southern highbush and rabbit for both years, and then rabbit was the highest in the second year. So what does all that mean? The results suggest that rabbit and southern highbush have a more desirable sweet-tart balance, that sugar-acid ratio, and northern highbush may taste more acidic, and this may be an indicator that rabbit and southern highbush have better fruit quality in terms of the sugar-acid ratio, or that sweet-tart balance, than northern highbush does. When we looked at how the individual sugar compounds broke down, fructose, this is the compound that's most related to how we perceive things to be sweet. In both years, southern highbush and rabbit were higher than northern highbush. And in the first graph, everything is correct. I just switched the colors by accident, so that's why it doesn't affect anything, but in case you're curious. In glucose in 2018, southern highbush was higher than northern highbush, and southern highbush was also the highest in 2019, followed by rabbit and northern highbush. And sucrose was so low, and it wasn't different between any of the types, so we didn't examine it further. So this may indicate that rabbit and southern highbush may be sweeter because of that, perceived to be sweeter because of the fructose than northern highbush, and may be more preferred by consumers. When we looked at the acid content, citric acid is often thought to be the most predominant acid in blueberry content, and it was for northern highbush for both years. Citric and quinic acids were the highest in southern highbush, and quinic and shikshimic were the highest acids in rabbit. So what does all of this mean? How did these taste, these sugars and acids, relate to consumer sensory perception? Well, we already found that glucose and fructose were our highest sugars. They were related. Interestingly enough, soluble solid content that you measure on a little refractometer, in our data, we did not find it to be related to sweetness, which is often thought to be the main way we think about, oh, how sweet is something? How high is the soluble solid content? We did not see that. Acid content was highly correlated with titratable acid. And interesting enough, none of the predominant acids that I had just mentioned in the previous slides were found to have any relationship to consumer preference. And consumer overall liking was highly related to flavor intensity, sweetness, and acidity. So let's go through, I gave you a lot of information, let's go through the main take-homes of that section of the talk. The results suggest that southern highbush ranked higher than northern highbush and rabbit for overall liking. And remember, the perception is that it's northern highbush, then southern highbush and rabbit. That's not what we found. So we found it for overall liking and flavor liking, southern highbush ranked the highest. And this is also my favorite, I guess if you can say favorite, but I found the most interesting result. The rabbit ranked the highest for aroma liking. So people are liking the flavor profile, that aroma from rabbit eyes. And again, that contradicts the perception of northern highbush being the highest quality. For summaries for flavor volatile compounds, how it relates to the consumer, the sensory perception, there were two chemical groups that were made up pretty much most of the volatile compounds, and the compounds in these groups contribute to sweet, floral, and fruity notes, also green and grassy aromas. The ones that contribute to sweet, floral, and fruity are the ones that have the highest relationship to sensory attributes from consumers, and maybe we can target those moving forward. For sugars and acids, again, soluble solvent content, not something that we found to be a perception of sweetness from our data set, and none of the predominant acids that we found in blueberry related to anything from consumer preference. But the perception of the sugar and acid contents is still influencing flavor acceptability. So the next part of the talk, the last part, this is just some preliminary work done by 2 of my graduate students, and we're still going through the last little bit, so not as complete as the first part. But basically, we wanted to look at how flavor is held up over storage. And it's thought that basically the longer that you have a fruit in post-harvest cold storage, the more influence that flavor will be. So it's supposed to degrade more than the textural traits or other traits. And also the concern is the development of off flavors and the loss of flavor. To date, there hasn't been anything that I've observed that's reported on this. So again, this is the same fruit. We got a lot of miles out of this fruit. Today, I'll just be showing 1 year and a subset of this data because, like I said, we're still finishing it up. So for this talk, we're going to look at 2 southern highbush varieties and 2 rabbit eye varieties. And we went ahead and then took them at different intervals. We did 4 post-harvest intervals. Time point 1 was 3 to 4 days after collection, so basically as soon as we got the fruit, then 10 days, 20 days, and 30 days. We looked at 11 flavor volatiles from those 2 main groups that we talked about, and we decided to hone in on 6 flavor volatiles, mainly because they were present in all of the cultivars across the time point, so we could get a good look at them. And we looked at compounds that contributed to the green grassy aroma, sweet and floral, citrus lemon, minty, and fruity flavor. So the results on this, so you don't have to pay attention to the trans-2-hexanol. I should have switched that and put the green grassy. So the first compound that makes that green grassy aroma in blueberries, for both types, no effect. So if you see like for Brightwell, for example, you see it starts with a B above the graph for time point 3 and ends with a B in time point 30, that means no change. So basically, rabbit eye and southern highbush, no change for this compound for green grassy aroma. For all of these compounds, there's something called an odor threshold. So if something is below the odor threshold, we as humans cannot perceive it. So it doesn't matter what changes are happening because we can't pick it up. Maybe a dog could or something, but we certainly can't. So camellia, any changes, everything was below the odor threshold, so that suggests that any changes in this compound wouldn't affect how camellia is perceived for taste. And it suggests that the green grassy odor does not change from this compound over time in the cooler. We did a second compound for the same green grassy flavor or aroma. In rabbit eye, again, no effect over time. Southern highbush, one of our varieties, it did so an increase in one compound. And again, this is a preliminary set, so these are not as solid results as the first part, but overall it suggests that this green grassy aroma does not change in rabbit eyes. Perhaps it is affected in southern highbush. For the sweet floral aroma, cold storage did not change for both rabbit eye and southern highbush, and it suggests that cold storage has no effect on these flavors. For the fruity aroma, for both types, all cultivars were below that odor threshold, so that basically means any changes in these compounds won't affect how we perceive it, that fruity flavor. So any changes over time, it's not going to really affect that fruity flavor, and also we can't perceive that. For limonene, the citrus lemon aroma, rabbit eye, it remained stable. In southern highbush, it varied. We saw an increase in one, a decrease in the other. And it suggests that citrus and lemon flavor might not change either. The last one, the minty, it did fluctuate over time in Brightwell, but remained stable in powder blue. And it wasn't even detectable in the 2 southern high bushes that we looked at. So again, the results suggest that it probably won't change over time, and any changes would only be perceived in rabbit eye. So the summary of that, the green grassy flavors do not change over time. Maybe it increases in southern high bush. The sweet, floral, minty, they do not change. And any changes to the fruity flavor, it may not have any effect on taste perception. So last slide, the overall take-home points from all of that. So this is the one slide to kind of think about. For flavor and consumer acceptability, results suggest that rabbit eye ranked higher than, southern high bush ranked higher than rabbit eye and northern high bush for overall and flavor, and rabbit eye was the highest liking for aroma. The majority of sweet, floral, and fruity compounds are what relates to consumer sensory perception. Soluble solvent content for our data set, not good indicator for sweetness. Over time, green grassy aromas do not change. Maybe it increases in southern high bush, and it doesn't change for the sweet floral, citrus lemon, or minty flavors. And fruity flavor, we have too low levels for it to be perceived. Lots of people contributed to the work and funding, and thank you, and I think I'm out of time. But I'm here, so if you have questions, please see me after.
Yeah, thank you so much. Thank you so much, Dr. Idle, for the presentation. Now we are going to continue with our next speaker, Dr. Luan Oliveira. He is an assistant professor at the University of Georgia, and he will be talking about AI application for quality predictions. Okay, thank you.
Thank you. So, good afternoon, everybody. Thanks for having me. My name is Luan Oliveira, as Lilian said. I'm an assistant professor at the Department of Horticulture at UGA. I'm based in Tifton. More than an assistant professor, and I'm a precision ag extension specialist. 75% of my time is dedicated to research for the growers. Here's my presentation title, AI application, artificial intelligence applications for blueberry quality predictions. So here's my team as far as January 2025. Without these guys, I would not be able to do all of this research. And if we talk about AI, there's a lot of buzz right now going on, right? It seems like 2 years from now, 2 years back, people talking a lot about ChatGPT and how AI is enabling a lot of things across agriculture. I could not start a presentation without talking about what AI is, which even if it's really basic, because I'm not an AI specialist, we just apply AI concepts into agriculture. So let's start with the basic concept. It refers to the capability of a machine to imitate intelligent human behavior. But the trick here is that you have to teach it. It doesn't learn by itself. People say that yet, so I don't know how long it's going to take. But the AI enable machines to perform tasks that require human intelligence, such as understanding language or recognizing patterns, solving problems, and learning also from experience. And I'm going to go a little bit deep into that. If we think about AI as a broad, it's not just AI, it's not just artificial intelligence. There are some subsections of that. And the computer guys at the Silicon Valley, they say that right now we have what they call weak AI. It's not strong yet. That's the AI we currently use for our projects, or that's what ChatGPT is. And then there are two more types, which is general and super intelligent. Those guys, they say that the general AI would be something in the future. It's really conceptual that this thing is going to be able to learn from itself. And then when the world reach a point of a super intelligent AI, that literally thing is going to be able to have emotions, like human emotions. but we are like really far away on that path, I'm pretty sure. So inside of our reality, we have like different subsets of the narrow AI, which could be machine learning. You could have learned have heard that some during your life, or generative AI, or computer vision. So inside of machine learning, which is something that we actually use for agriculture on robotics, or on predictability, forecasting, we have two types. We have supervised and we have unsupervised, and that's really funny because both of them are in our daily lives, and we didn't even realize. So supervised AI is something that you teach, it's labeled. You inside of a cotton field, you teach a robot to find a apple. So you just tell the robot, hey, an apple is round and red. So that artificial intelligence is going to find those patterns and find the apple for you. But the unsupervised is kind of behavioral. So I'm pretty sure most part of us here, we watch Netflix or Amazon Prime or any other platform, that sometimes you like some sort of movies, and that AI kind of brings that to you. It is on your first page there. It says, you may like this. That's unsupervised AI. If you watch a lot of action movies, that detects your behavioral taste for movies and say, hey, you might like this, or a shoe, or something like that. So that's machine learning and unsupervised artificial intelligence giving you those informations. And then generative AI, ChatGPT, is on TV, is everywhere. You say, hey, Chat, give me a 30-minute presentation for Blueberry Public. So that's what I'm presenting here. I'm kidding. It gives you that. And then computer vision, robotics. In 2024, there are more than 400 companies related to robotics for agriculture. And this number was only 200 in 2022. So this is becoming more and more and more a reality. Okay, and those are only 3 subsets. We have more. There are at least 10 more subsets of AI that can be applicable to agriculture, for example. So narrow AI in ag. So I listed some applications that can be done through AI in agriculture. I'm not gonna read through everything, but you see robotics and automated machinery. So the machine learning models would enable ground vehicles to take inside decisions. For example, remove the weeds or spot spraying, things like that. And then analyze data, from sensors, from drones, from satellites, and then do monitoring, giving you feedback after that analysis. But there's also, what I'm going to present here, something that we do predictions. We forecast how blueberries is going to be in 10 days, for example. And then we cannot do it without remote sensing. So this is another topic. This is not AI anymore. This is what we do since 1990s in agriculture with drone or satellite images. As the name says, remote sensing, you collect a data set without being there. If it's not remote sensing, it's going to be proximal sensing. Let me see if I can put a pin here. Can I? I don't see my mouse, right? You see a human being just below the drone, so that's proximal sensing right there. We use these sensors, cameras, which are phone cameras, like RGB, like normal cameras, or multispectral, what we call. Those cameras, they capture things that we can't see. And I'm going to go a little bit further on that. On remote and proximal sensing, you have 2 different approaches, okay? You can use a spectral response, which is the reflectance of the blueberry bush, for example, and that gets a little bit deep here. But If this room was really dark, right, like the absence of light, we could not see anything. Is that right? We cannot see it because what we see is the reflection of the light in the objects. Our eyes, they're sensors, so they capture the light in certain wavelengths, and then we're able to see. That's a fact, okay? That's science. And then one way of remote sensing is capturing that reflectance, and the other way is identification and segmentation of objects. How we do that? Let's see here. This is a beautiful morning right there, and the sun is rising. So the sunlight touches the leaves, and the chloroplasts inside of the plant cells, they're going to reflect, because they have a lot of chlorophyll, they're going to reflect that green, and we're going to be able to see green. But when the plants are stressed, we don't see green. Why? Because there's no chlorophyll there anymore. So that light, it can be absorbed, it can be transmitted, but it also can be reflected. So that's our catch right there. When we, that's just a graph right there showing that there's what we can see and what we can't, right? So inside of the light spectrum, we only can see in between 420 and 740 nanometers. And after that is infrared, and below that, ultraviolet. So we use spatial sensors. Why? Because if we think about a healthy, a stressed, and a dead leaf, I want you to look just at the NIR, the near infrared. The plants, they reflect a lot of near infrared when they're healthy. But when they're stressed or dead, it doesn't matter. But the red matters. So here's a catch. We can calculate something called NDVI, which is nothing more than a vegetation index. That's going to tell me if that plant is reflecting a lot of near infrared or a lot of red. I call as a difference is the near infrared minus the red divided by the near infrared plus the red. Okay, and then I have NDVI, which it goes from -1 to +1. -1, I have more red. +1, I have more near infrared. +1, my plants are healthy. -1, my plants are stressed or dead, basically. And then this is the spectral response approach. And then when we go to identification and segmentation approach, that's how we do it. And this is AI already. This is called machine learning. We, here in this video, and that's actually not, my student did it. We took pictures of berries right there. All of them were ripened. But we taught the computer to say, hey, only identify objects that are more blue than white, or within this color range. And then the model learns from what you thought and say, okay, Luan, I'm going to just get those berries for you. And as a plus, the computer also gets the colors. It also collects data in the red, green, and blue range, what we can see, because in this case here, we only used a phone camera. And that's how it goes for our study. So this is a study which is under review right now. He's not here, but Mr. Mike Brewerton, he was nice enough to give us a lot of berries, 3,600, thank you. And then we could make this study happen. We collected, we harvested 4 times 45 bushes of blueberries, Southern High Bush this year. And from that harvesting, we used those ripened berries to train a machine learning model to use our phones to take a picture of that, of those berries. And we want to see if only using the phone, we could say if one, that berry had high TSS, total soluble solids, and then a lower or higher firmness as well. These were 2 questions that we wanted to answer. So with that, we got 3,600 berries. We took pictures of every single one of them inside of that little studio right there. And then with a phone, like an iPhone. And then we use this artificial intelligence model to identify and segmentate those berries one by one. And then we extracted the data. You see there red, green, and blue values, the RGB values from those berries. And then here are some results. So we got the first harvesting at 114 days, the second one at 123, third one at 134, and then the fourth harvesting at 142. Interestingly is that at 123 days, we got more color intensity, red, green, and blue color on those berries, and we're still analyzing that data. And then we brought those to the lab. There was not only pictures, of course, because for training an AI, you need the actual ground truth data. You know, your model is going to find patterns inside of the of your data set and tell you, hey, in this color, your blueberry is more sweet. In this color, your blueberry is more firm. So this is what we wanted to do. So we use the spectrophotometer to measure the TSS and the space color index, and then a penetrometer for firmness. And then we have some results there that I'm going to skip. Do I have time, Lillian? How much time do I have? Okay, so I still have plenty of time. Okay. So, and then you see like during the 4 harvesting times, we also found some patterns that at 134 days, the firmness of those berries were higher at the 4th harvest time. And that was weekly, okay? At 114 days, they were a little bit bigger in the first harvesting. And then we could use those inputs to train the artificial intelligence model on, for predicting those variables. We found an issue in the way. Our data set was only composed of ripen berries. And there's a catch there that when you're trying to train an artificial intelligence model, you need variability. You need to, from green to ripen to unripen, you know, midway, you need a lot of variability there, and we did not have that variability. So we used what we call a Bayesian general linear model to create an artificial data set based on on the data set we had. So we had 3,600 berries there, and with this model, we increased that data set by 1,000 times more. So you see here, there's an observer curve. This is our data set for TSS and firmness. And then those yellow curves that you see right there, they are the simulated data sets for creating the AI model. And we found some correlations in between the RGB, the cell phone images, with the quality parameters, the diameter, the firmness, and the TSS, also with the space color index. And for us, you know, what the results were showing was a no-brainer. As green the fruits were, more firm they were. As ripen the fruits were, like as bluish the fruits were, more sweet they were as well. And that happened on all 4 harvesting times we got. So, but what we found also here is that in the first harvesting, the 3 principal components that we found were only explaining 75% of the data. And along the harvesting, the second time it went to 84%, the third time it went to 90%, and then the 4th time it went to 91%. And we found 3 main principal components inside of the data set that would explain the whole data. So then we generated the artificial intelligence model. We applied those models to our data set to see how much we can predict blueberry sweetness, TSS, total soluble solids, using different models. So here, each square that you see on the screen right there is a different artificial intelligence model, multiple linear regression, decision tree, support vector machine, random forest, a lot of different models. And then we found that the multiple linear regression was better to predict at 0.99% confidence using phone images, the TSS of the blueberries. And then in second place, we would have the support vector machine, and third place, the artificial neural networks. So this is a TSS, and then when we go to firmness, pretty much all the models that we tested were telling us that we could predict that using the phone at over 90%, with over 90% accuracy. And I just brought here two different explanations on how basic machine learning models would work as a decision tree. There is a game on Alexa that you kind of say, hey, Alexa, guess what I'm thinking? And then Alexa starts saying, is this a soccer player? Is it, and then it plays for Barcelona, and then till it reaches Cristiano Ronaldo. It's something like that. You say, hey, Alexa, what is my blueberry sweet or not? And then it says, is the average blue over 150? And then, yes. Is the average red lower than 100? And then if yes, it's going to be firm, or vice versa. But there are more complex models, and this is a decision tree. It's a really basic model, but then a random forest would have a lot of those trees. So that's why, we don't do by themselves using the pen. We use computers to do that for us. Some next steps on this research would be to develop and validate an app, like a phone app, or a platform on a grading machine that would run those models in real time for blueberry quality assessment. And of course, scale up for packing and sorting facilities, and implement those models into robots for intelligence harvesting decisions. I prepared my presentation for 20 minutes, so I think I'm on time on that, Lillian, and I'm open to questions right now. Thank you. We do have data on drones, and my postdoc is bringing right now some handouts on that. But on stress, that's basically what I explained in the first couple of slides right there. If we fly a drone and if the NDVI is higher, the plants are not stressed, and if that NDVI is lower, the plants are more stressed. That's basically how it works. So we don't have like a direct result, but we can do it. Yes, sir. Yeah. Go ahead. The question was if I found where the RGB values are really close to the ground truth value. Is that right? Yes, because yes, we did find some correlations with that, and that's on that correlation matrix right here. You see that the blue would correlate with firmness, actually all of them, the red, green, and blue, positively with firmness of those berries, and negatively with the TSS. Those were the results we got. Thank you. That will be good. So that's what actually we want right now, is to apply this model on different varieties. I'm sorry I didn't say this was a southern high bush farting variety. So we want to apply the same model to different data sets to see how is the fit of this model, if it's going to work on, like a rabbit eye, blueberry as well. Or we're going to have to train all of these models for different varieties. Did I answer your question right there? Okay, thank you. You had a question, right? Okay, thank you. We like this year, actually, we're going to harvest the same field, and we're going to do a more variable data set to strengthen the model, like to tell the AI that, hey, there are not only ripened berries, there are unripened as well. Thank you, sir. Thank you. Oh, there's a gift. Thank you.
Thank you so much, Dr. Oliveira, for the presentation. Now, we are going to continue with the presentation of Natalia Espinosa. She is going to talk about biochar. She is a PhD student at the University of Georgia.
Thank you, Lily, for this presentation. Good afternoon, everyone. My name is Natalia Espinosa. I am a graduate student from the University of Georgia. I work with Dr. Silfina Rubio in the small fruit lab. We are working right now, I am working right now with using biochar as a soil amendment in blueberry production. These are some preliminary results from my research. So this is from only 1 year. So first of all, here in Georgia, we work with, as everybody said before, 2 types of blueberry. We have the southern hibiscus and the rabbit eye, and they are both have low chill hours between 200 and 600. And as any other blueberry, they like the pH of the soil low, like between 4.4 and 5. And this is important because I'm going to be talking about soil a lot. So again, here in Georgia, our soil is sandy, where we produce blueberries, is sandy or sandy loam. And this is important because as it is sandy, it has low soil organic matter, low cation exchange capacity, low pH buffer capacity, and for these reasons, we have to add soil amendments, such as pine bark. So before I go further, I just want to explain these concepts a little bit better. The cation exchange capacity is that the capacity of the soil to attach the cations, such as calcium, magnesium, and ammonium. And this is important because when a grower adds a fertilizer, you want the fertilizer to stay in the soil. So you don't want the rain to run off all the fertilizer. So you need cation exchange capacity to be higher in your soil. The cation exchange capacity is higher when your soil has high soil organic matter. And the soil organic matter comes from soil amendments such as biochar, such as pine bark, and also comes from the leaves that the plant itself, during the senescence, it let it fall on the ground. So, and also it comes from clays in the soil, but we don't have a lot of clays here in our sandy soils. That is the part of the texture that don't play good with us. So because of that, we have to add soil organic matter to our soil to increase the cation exchange capacity and to maintain those nutrients in our soil. But not only negative sides comes with the organic matter, we also have positive sides. And to these positive sides, we have these anions as phosphates to be attached to. And this is important again because we also apply phosphates and we don't want these phosphorus to be lost. The problem that we are seeing here in Georgia is that our soils, we apply a lot of phosphorus, but the plants are not taking up that phosphorus to themselves. And this is because our soils have the capacity to fixate the phosphorus so well that the plant cannot uptake it. And then we also have this, the pH buffer capacity. So because our plants, our blueberries like low pH in the soil, we want the soil to remain in the same range of pH. But when, again, we add fertilizers, this pH tends to be lower and lower after many years of application. In the field, we have seen farms with pHs as low as 3. And this is a problem because when our pH in the soil is low, we have less attachment to these nutrients, and we have the release of aluminum and iron to the soil solution. And the plants start to uptake this aluminum and iron, and at some point it can be toxic to the plant. So after many years, 15 years of planting blueberries there in the same spot, adding fertilizer year after year, you're gonna have a pH in the soil very low. So you want your soil to have high pH buffer capacity in order to avoid the soil to go low. Also, it avoids the soil to go up. So again, we are adding this pine bark as a soil amendment to increase the cation exchange capacity, to increase the soil organic matter, and to increase the pH buffer capacity of the soil. But these properties are going to be lost with time, because with time, the pine bark tends to decompose. But the growers apply pine bark every year in order to try to maintain the pine bark. But again, you're applying it on the surface. You cannot integrate it in the soil as you do at the beginning when you're establishing the field. So this is a problem again. So our theory is that we need a soil amendment that lasts longer in the soil. So this is the reason we are trying with biochar. We want a soil amendment that lasts longer so it decomposes less. The secret is that it decomposes less because it's less available for the bacteria to get into the carbon in the biochar. So we are expecting that this soil amendment is going to last longer, and it's going to increase these characteristics that we want in our soil. Okay. That is, I just wanted to explain that the pH is the quantifier of the hydrogen, the protons that we have there, and then when we have more positive and negative sides, our soil is more able to support, to avoid the increase or the decrease of the pH. So, as I say, we are trying to this biochar as a soil amendment, and you can see here that it has very good properties. It has all the nutrients that you see here. It has high quantities of phosphorus, potassium, magnesium, calcium when compared to pine bark. And the good thing also, you see the pH is 6.3. The problem with biochar that we have seen in other trials is that the pH of the biochar can be very high. And this is a problem because it can increase the pH of the soil. So we are trying, our biochar has a pH of 6.3. I know it looks high, but it's not as high as other biochars that we see that has pHs of 9. So this is the biochar that we are using. And to try this theory, we have 4 experimental plots around Georgia. We have 2 commercial farms in Alma. We have 1 research farm in Alapaha. And we also have a greenhouse experiment. In the 2 commercial farms, we applied 3 soil amendments. So we have, in the farm 1, again with farthing, we have 33% in volume of pine bark, and we are also trying 9% in volume in the soil of biochar, and also none, without any amendment. These amendments were incorporated into the bed, as you can see in picture 3, before planting the blueberries. In farm 2, the one that is in picture 2, where I am, so that farm has an Optimus southern highbush. And so for this farm, we had 2 soil amendments, 33% in volume of pine bark or 9% in volume of biochar. And these amendments were incorporated into the whole because the plants were already there when we started this. So we also are trying 2 different types of amending the soil, one incorporating everything or just applying into the whole after the plants are already there. And then in the Alapaja Research Farm, we have 4 soil amendments. One, the 33% pine bark, one 9% biochar, another one 20% biochar, and also none. Again, they were incorporated into the bed, and is the picture that you see in image 1. And then we also have a greenhouse experiment with pots. This greenhouse experiment, we established it because we wanted to see how the biochar behaves with the rain, do artificial rainings on our greenhouse to see the leaching content and see if actually the biochar is retaining those nutrients in our soil. So our results, the first thing that we want to see is the pH buffer capacity. As I told you before, the pH buffer capacity is the capacity of the soil to maintain the same pH. And we divided this capacity between if the soil is less prone to reduce the pH or if the soil is less prone to increase the pH. So as you see in the table, it depends on the time of the application of the soil amendment. So it changes with the texture of the soil, it changes with the farm, it changes with the time of application. What we are seeing until now with this is that at the beginning, both of them, pine bark and biochar, are behaving well, supporting the pH buffer capacity of the soil. But with time, the pine bark, both are losing that capacity. Both are losing the property to maintain the pH buffer capacity. So probably we need to try different other rates to see this improvement here. The same with soil organic matter. It depends on the texture of the soil and the farm. We see mixture of results here. As you see, for example, in the greenhouse, we see that both soil amendments behave well and increase the soil organic matter. But then we see that in the Alapaja Research Farm, the last one, that the pine bark increased the soil organic matter, but the biochar at both rates didn't increase the soil organic matter, actually decreased it. So it depends on the texture of the soil and the time. And as I say, these are preliminary results. We'll see how they behave after the second year. This is also part of what I am explaining right now. So you have in the axis, you have the soil pH, the cation exchange capacity, and the organic matter after from the baseline, 6 months, 14 months, 18 months, and 19 months. And I just put this graph here because I want you to see that, for example, we have significant difference between the soil amendments when we are talking about pH. The biochar has higher pH at 14 months, but after 19 months, we don't see the difference anymore. So with time, the biochar, the pH in the soil is stabilized, and we don't see actually a change in the pH between soil amendments. So that is good because we don't want our pH to go very high. And then for the cation exchange capacity, we see that at the beginning, again, the pine bark is higher, but with time, it is stabilized. So we don't see a significant difference. And then we see the same with the organic matter. So kind of they start behaving the same with time. In the farm 2, we saw that pine bark amended soil showed higher concentrations of phosphorus, zinc, and manganese and copper than biochar amended soil. So that is what you have in green. This is after 19 months of application. In the Alapaja Research Farm, we saw that the biochar at 9 and 20% increased soil pH, as I say at the beginning, but with time it stabilized. And then we also saw that the biochar at the rate of 20% volume increased the cation exchange capacity when compared to the other soil amendments. That's good, that's what we expected. And one thing that I noticed is the cation exchange capacity actually tends to increase in all the soil amendments, but it increased more with biochar. And the soil organic matter was not significantly affected by any soil amendment. And this is a problem in Georgia because it doesn't matter what kind of soil amendment we are adding to the soil. We've seen farms that don't go not even close to 3% in soil organic matter. And again, the blueberries need more than 3.5. So this is a general problem. Also, in the case of the Alapaja farm, for the soil nutrients, Here we have good results with biochar. We see that biochar had higher levels in the soil of potassium, magnesium, calcium, boron, zinc, manganese, copper. As you see, the pH of the soil was also higher, but not as different. It's not very high. It goes until 5.4, blueberry support until 5.5, and with time it tends to decrease, so we're expecting to see that in the next year. Another thing that I want to point at is that the soil that didn't receive any amendment had a higher amount of iron. You see, because we don't have organic matter, we are seeing more iron in the soil that probably can go to the toxicity of the plant. So just to point out the importance of organic matter and the soil amendment. In the case of the plant growth parameters and leaf nutrients, so we measure the plant height, the cane diameter, the leaf area index, the chlorophyll spot, and just to summary of everything, we didn't see a significant difference between soil amendments in any of the farms. Actually, we saw a negative for the biochar in Alapaja, for the cane diameter, but not for anything else. We did see a negative result for the 9%. So we are seeing here that actually it's better to use 20% rate of biochar than 9%. And for the greenhouse, the leaching part, it's important to mention that the biochar amended pots are leaching more boron, magnesium, and calcium, probably because the biochar had much higher amounts of these minerals than the pine bark. But the other important thing is that when we have pine bark, we are leaching more aluminum, phosphorus, and manganese. And phosphorus, as I said before, is very important because the problem with our soils is that we have phosphorus in our soil at the point of leaching, but the plants are not taking up that phosphorus. So we have to keep seeing this and see how we can improve these conditions, change the rates for future research, and see how it goes. The other interesting thing, we are also measuring the colonization of mycorrhizas. The baseline, after 6 months of applications, we didn't see a significant difference. We are going to measure again this year and see if we have any difference between the biochar and the pine bark on the pots in the greenhouse. By now, after 6 months, it's just too early to see. We didn't see a significant difference. And that's all that I have for now. If you have any questions. Also, I have to thank to the Georgia Department of Agriculture that they funded this research, and also my team in the lab that are always helping me. Sorry, can you say that again? Definitely. I mean, the phosphorus in the soil is detrimental for the environment because, as you see, it's leaching right now with the soil amendment that the growers are using. The pine bark is leaching phosphorus and is detrimental for the environment. But for the plant, I mean, the plant is not using that mineral, it's not using that phosphorus. And also, when you have too much phosphorus, it doesn't leach alone. Sometimes it leaches also with nitrogen. It creates alliances with nitrogen and potassium. So you're losing not only phosphorus, but all the other minerals as well. Yeah, it's what we're seeing. We see farms with pHs, soil pHs as low as 3. And the problem is that it is bad because the iron and the aluminum start to, we have the minerals in our soil have high aluminum and iron. That is the way of the soils here in Georgia. But when you have low pH, those minerals start to get soluble in the water that the plant uptake, and that is the problem. Thank you. No more questions? Gerardo. Thank you. Yeah, that is probably could be a problem because, as you know, it has to be in balance with potassium and other minerals. So that's what we are going to see, I guess, this year. We have to measure the calcium, but the good thing about the calcium availability in the soil when we have low pH is that it's not very soluble for the plants to uptake. So probably it's going to be in the soil, but it's not going to be uptaken by the plant if it doesn't need it. Percent? If you have higher than 3.5, you should be good. Okay.
Thank you so much. Thank you, Natalia. And following our programmation, we have a 10 minutes break, 12 minutes break. So we will continue at 2:50. Okay, we are going to continue with our next speaker. We are going to welcome Joy Ganciorek from the University of Florida. He's a PhD student, and today he's going to talk about biology and management of blueberry. So thank you so much, and the floor is yours.
Okay, perfect. Hello, everyone. I know it says Oscar Liebert up there, but I am not Oscar Liebert. I am Joey Ganciorek. I am a PhD student in Dr. Liebert's lab, and today, on his behalf, I'm going to be talking with you about the biology and management of blueberry gall midges in southern blueberry. So I'm going to start by giving a quick outline of today's talk. I'm going to start by giving a background on the blueberry gall midge, including biology and damage. I'm going to talk about gall midge phenology and how that plays into pest management. I'm going to talk about monitoring for the blueberry gall midge, including how we can enhance that monitoring using pheromones. And then finally, I'm going to talk about the management of midges using reduced risk and conventional pesticides, as well as how those chemicals affect natural enemies such as parasitoid wasps. So the blueberry gall midge is endemic to North America. It was first detected in Mississippi, but it is also well established in Michigan and many other blueberry producing states throughout the Northeast and Southeast. It has also since moved into the West Coast, into areas like Oregon and Washington through the transportation of nursery plants, and it can also occur as a secondary pest in certain areas of Canada. Overseas, it can also be found now in Europe. It was first detected in Spain starting in 1996, and more recently, it has also been found in Asia in countries like Japan and Korea. So here is the blueberry gall midge and its life cycle. So the adults are really minute pests. They're only like 2 to 3 millimeters in size, and they're orange in color. The females will lay their eggs on developing leaf and flower buds, and they might lay multiple eggs in a single go. After 2 to 3 days, the eggs will hatch and the larvae will begin feeding wherever they emerge from. Once the larvae are fully grown, they'll actually drop off the plant and onto the ground and then pupate inside of the soil. Once in the soil, they'll remain there for 5 to 10 days before emerging as adults. Altogether, this life cycle can take anywhere from 2 to 4 weeks, depending on environmental conditions. So, as I mentioned before, the damage caused by the blueberry gall midge is primarily caused by larval feeding. So, feeding on developing leaf and flower buds will cause a sort of burnt and crumpling appearance at the tips of these tissue. So, you can see an example of the damaged leaf tissue here on the left, and the flower tissue on the right. At some high enough infestations, it can cause the entire flower bud to die off, and then for leaf damage, it can cause severe stunted growth on the plant as well. Some growers have even reported that damage from this pest has resulted in up to 80% yield loss within their field, so it has a high propensity to cause damage as well. So again, the blueberry gall midge is a very significant pest of blueberry production within Florida and other blueberry producing regions in the country. In fact, back in 2018 and 2019, the blueberry gall midge was actually considered the most destructive pest of blueberries in Florida. with up to 58% of farms reporting heavy infestations during those years. So clearly, this is a pest that has a very high likelihood to cause lots of damage. Now, the main way that this pest has been managed is the use of conventional broad-spectrum insecticides. And while these chemicals can be highly effective and inexpensive, they can also cause damage to our beneficial arthropods, things like pollinators and natural enemies. So what our lab has been trying to do is trying to identify reduced risk pesticides that we can use that also work to conserve our beneficial arthropods, as well as other alternative strategies that may reduce the amount of spraying that we need to do in the field, which would also help to conserve our beneficial arthropods. So the first thing we investigated was the diurnal or daytime activity of the blueberry gall midge. So remember that the larval stage, that damaging stage of the blueberry gall midge, is inside of that developing tissue. So when we try to use something like a contact-based insecticide, it's not going to really do much because they're pretty protected. But we also know that at some point during their life cycle, they're going to have to leave that tissue to go into the soil and pupate. And this provides us with a good opportunity to manage them. If we can figure out the time of day in which this midge is going to move into the soil, if any such exists, then we can use that as a window to target this very vulnerable pest stage. Similarly, for the adults that eventually have to come out of the soil, we can also try to figure out the day in which they're most likely to come out of the soil, and that's going to provide another window for potential management opportunities. So what we did is we conducted a field study at a southern highbush blueberry planting in north central Florida. We set up the study following a randomized complete block design, which you can see pictured here on the bottom right. And we tested 4 different treatments, which consisted of 3-hour intervals. So we had a 6 to 9 a.m. interval, a 9 to 12 p.m. interval, a 12 to 3 p.m. interval, and a 3 to 6 p.m. interval. So as an example, say the 6 to 9 a.m. interval, we would put a sticky trap out starting at 6 a.m. and then collect it again at 9 a.m. to see everything that came out during that time. Each of these different treatments was replicated 6 times. So the actual trap itself consisted of a sort of white bucket with the bottom cut out and replaced with the sticky trap. So the soil was exposed just underneath that sticky trap. So adults that were just underneath the trap in the soil would come out and fly up and get stuck at the bottom of the sticky trap, whereas larvae that were falling from the bush trying to get to the ground would get stuck at the top. In addition to that, we also used HoboWare data loggers to get the environmental conditions. So here are the results, starting with the adults. So this first graph here at the top shows the mean number of adults captured for each of our different time intervals. Each time interval is also further broken down. So if you look on the left, you'll see these blue bars that represent the males, and then on the right, you'll see the pink bars that represent the females. And what we see here actually is that the peak emergence is different between males and females. Males were most likely to emerge between 9 a.m. and 12 p.m., whereas females were most likely to emerge between 12 p.m. and 3 p.m. Now, there was a lot of similarity between those first 3 time intervals, especially in terms of male emergence, but we really see things start to die off towards the end of the day from 3 p.m. to 6 p.m. In addition to this, we also looked at the environmental conditions close to the soil. And what we see is that with this orange solid line here, that's the temperature. We see temperature slowly increasing throughout the day. And then the relative humidity, that's this blue dashed line, slowly decreases throughout the day. And I'm going to get back to the importance of that in just a second. So next, we have the mean number of larvae that we caught per trap. And with the larvae, what we actually see here is that most larvae are coming out and trying to get into the soil from 6 a.m. to 9 a.m. So this is very different than the adults that were starting to come out around the mid-morning. But when we look at the environmental conditions inside the bush, we see very similar trends. So we see the temperature also steadily increasing, and then the relative humidity steadily decreasing. So what this tells us is that because these environmental conditions are the same or very similar between the bush and the soil, the difference in responses in the larva and the adults is more likely due to the inherent nature of those life stages and not because of the difference of environmental conditions between the bushes and the soil. So again, just to summarize this study, so the adult emergence peaked from the late morning to the midday. This could be potentially useful for monitoring purposes, but in terms of spraying, it's probably not going to be a good time to do that. And the reason for that is that a lot of beneficials, particularly pollinators, tend to be most active during this time of day as well. And we want to try to avoid impacting them. But the larval emergence time early in the morning could potentially be a good window in which we want to try to spray for them. Again, the larvae are very vulnerable, and the beneficials that we're trying to conserve tend to not be as active during this time of day. So for the next thing that we wanted to look at is we wanted to look at the use of pheromones. So blueberry gall midge females do produce pheromones in minute quantities to help attract male mates. And the use of pheromones could potentially be utilized for pest management purposes. One of the main things that this could be used for is enhancing the attractiveness of traps that we're using. A lot of times we're using clear sticky traps to try to catch these pests. So on their own, they're not very attractive, but with a good lure, they might get a better catch rate. And it's important to try to get this high catch rate because it's really useful for getting a better understanding of the population dynamics of this pest. So we can better see things like where hotspots are occurring in the field or when their population is starting to ramp up. And another important thing is trying to get early detections of a pest. The sooner we get a detection of a pest in the field, the sooner we can get to management, and that greatly decreases the chance that we're going to get significant infestations. There's another potential application for pheromones called male mating disruption. And with this, we kind of inundate the whole field with pheromones, so much so that the males can't really properly identify what's a real female and what's the lure. That decreases the amount of mating that occurs in the field, and therefore decreases how many midges come out in the subsequent generations. So for the first study we wanted to look at for this was the use of pheromones for just enhancing the trap capture. Are we going to get any sort of response in the field from these? So for this, again, we were in a southern highbush blueberry planting in north central Florida. We used a randomized complete block design, but we only had 2 treatments. We had traps without the lure and traps with the lure. And the lure itself consisted of a pheromone lure provided by Russell IPM based out of the UK. And what we did is we took this pheromone and we put it in these little rubber septas, which you can see pictured there at the top. So these little septas are able to hold much more pheromone than the female blueberry gall midge could produce on its own, and they also provide a good slow release effect so the pheromone's not just going to dissipate after a few days. Oh, and I'll also mention that, so we were using the clear sticky traps to catch the midges, and we did this weekly over a period of 5 weeks. So the results, this graph here shows the mean number of adults captured between males and females, and then those 2 sexes are further subdivided between baited and unbaited traps. So what we see here is that for both males and females, there's significantly more adults being captured by the baited traps than the unbaited traps. But we do also see that the males have a much higher relative increase when we use those baits. The amount of males captured increased by 116% when we use these pheromone lures. And this isn't really that surprising since the pheromones are for attracting the males. So it's not really too surprising that the males were more attracted than the females were. But it's still good to see that the females did have some sort of response as well. So this next graph instead breaks down the amount of midges that we captured over time. So our 5 different weeks we were collecting data for. This graph is also just for the males. So what we see with this graph is we see 2 different things. So the bar that is mostly on the top, the orange one, is the baited trap, and the one at the bottom is the unbaited trap. So what we can see here is that the population of males peaks at weeks 2 and 4, and this is significantly higher than weeks 3 and 5. We do kind of see the same trend with the unbaited traps, but there's not a significant difference there. And so what this means is that these baited traps are much better at getting this population dynamic, this change in the population over time, which can be really useful for finding things like when the infestations are really picking up or when there's hot spots in the field. In addition, we also see some significant differences at specific time points, most importantly at week 1, where there's significantly more being caught in the baited trap and no midges being caught in the unbaited trap. And this is also really important because now that we see that we caught the midge right at the start here, that might potentially inform the grower that maybe it's time to start doing management, whereas if they use the unbaited trap, then they might not think that it's time to start managing the pests. So, again, the key findings from this study is that the pheromones can increase the trap capture rate of males and females, though we do see a stronger response with the males. And then we also see that the baited traps are better able to capture certain population dynamics compared to the unbaited traps. So the last topic that I want to cover for today, and this might be most interesting for some of the concerned growers in the room, is that we want to try to figure out what are the most effective chemicals for blueberry gall midge. In addition to that, we wanted to see how these chemicals are affecting a key natural enemy group, parasitoid wasps. So parasitoid wasps can provide significant natural control over blueberry gall midge, and they have the added benefit is that they're targeting a specific life stage that is really hard for the pesticides to reach, which are the larvae. So we want to try to do our best to identify not only chemicals that are really effective against blueberry gall midge, but also try to find ones that can conserve natural enemies. So again, we conducted this study at a commercial blueberry planting in north central Florida, following a randomized complete block design, and we used 10 different spray treatments. So here is a big table of the logistics of all of the different spray treatments they used. Just to go through very quickly, we had Asail or acetamiprid, Exarel or citraniliprole, Savanto or flupyradiferone, Admire Pro or imidacloprid, malathion, Delegate spinatoram, Movento spirotetramat, Movento plus Induce, which is an adjuvant, and then Apta or tolefenpyrad. And on top of that, we also had an untreated control as a comparison. Going back here again, so each plot consisted of 5 blueberry bushes. Each would be treated twice over the course of the study. So we did 1 spray, and then 2 weeks later, we did the second spray. All sprays were applied using a CO2 backpack sprayer, which you can see pictured here on the right. And all sprays were conducted either early in the morning or early in the evening to avoid damaging any key beneficial arthropods. For sampling, what we did is we did 2 types of sampling. We did destructive sampling, so this involves collecting flower buds. We did this 2 and 8 days post each spray treatment application. So that's 4 samples total that we collected. When we collected the buds, we collected 25 from each treatment plot, and then that would lead to 100 total flower buds per treatment. We put these flower buds inside of an incubator and then counted how many larvae came out. For the adults and parasitoids, we used clear sticky traps. And again, we did this, collected them 2 and 8 days after each spray treatment. There was a single sticky trap per plot for a total of 4 traps per treatment type. So, the results. So this graph here shows the mean number of adults that we captured for each of our different insecticide treatments. And what we can see here is most of the insecticides that we tested, so from Asail going on to the right, they all provided significant control over the blueberry gall midge. But 3 of the chemicals performed the best. So we have Moventa with the adjuvant, Apta, and Admire Pro, providing reductions of 68%, 59%, and 37% respectively. Next, we have the data on the larva. The insecticide sprays weren't quite as effective on the larva. Very few of them provided significant reductions. But again, this isn't too surprising since the larvae are inside those developing leaf and flower bud tissues, so they're relatively protected. The 3 that we did see a significant effect are were, once again, the two most effective we saw for the adults, which were the Movento with the Induce and Apta, providing 56% and 51% reductions respectively, as well as Sivanto, which provided a 33% reduction. And then finally, we have our parasitoid wasp effects. So I won't go into the details. We did break it down quite a bit by different taxonomic groups, but for the purposes of this talk, I'm just going to focus on the overall abundance of parasitoid wasps. So what we can see here is that for almost all of the different chemicals, there is a notable numerical reduction, but the numbers are not significantly lower compared to the control. So that means most of the chemicals that we tested, including our two most effective chemicals, the Movento with Induce and Apta, were pretty good for conserving these beneficial parasitoids. So just to quickly summarize this last study again, Movento with Induce and Apta seem to be the best options for managing blueberry gall midge because they were able to effectively reduce both the adults and the larvae. In addition to that, they also did a pretty good job of conserving parasitoid wasps, which might provide more downstream effects in terms of control. And then finally, while most of the chemicals we did test did significantly reduce the adults, there is still a need to try to keep looking for new chemicals that can attack the larvae because these results didn't find too many. And with that, we are at the end. So I just want to take a quick second to acknowledge a few folks. Everybody listed here significantly contributed one way or another to the research. I also wanted to give a shout out to FDAX for providing funding for this work. And most importantly, I want to acknowledge Dr. Marise Lopez. She's pictured here just to the right of the long list of names here. Marise is the former member of our lab who actually conducted all of this work on blueberry gall midge and more. She's an all-around amazing and intelligent person, and I'm really grateful to have had the opportunity to share all of her work with you here today. And with that, we are at the end. Thank you so much. Yes. Could you repeat that again? It was 2 and 8 days is what we did for collecting. From what I can see here, no. Again, this isn't the work I conducted, so I have a limited pool of information available, but I'm assuming not. Yes. Well, I don't want to state too much because, again, this isn't my work, so I don't feel like I'm the most qualified. Based off the work I presented here, it looked like Movento and Apta did a good job. But again, I'm not an expert on this particular system, so I don't want to say, like, use Movento, use Apta, and then it doesn't work out for you. But based off of the data that I'm showing here, it seems to work reasonably well. Mm-hmm. Yeah, absolutely. And again, when we were doing these applications, we did them early in the morning or late in the evening to try to avoid hitting those pollinators. But definitely, and especially with the blueberry gall midge, by the time they're already starting to flower, I mean, obviously there's going to be some overlap because if you have cultivars, one might be flowering, one might be budding still. But by the time they start flowering, the gall midge is probably already moved on at that point. Yes. Oh. All right. Thank you, everybody. Thank you. Mm-hmm. Oh, thank you.
Thank you so much, Joy. Now we are going to continue. Our next speaker is Dr. Elizabeth Canales, an associate professor and extension economist at Mississippi State University.
Good afternoon, everyone. Thank you for being here today. Thank you for the invitation. So I'm going to be talking today about two research papers looking at consumer preferences for blueberries. And this is, first I want to acknowledge some of the, all of the co-authors in this these two research papers. So this is part of a USDA-funded project and backup project that Massimo Iorizo talked about earlier this morning. And one of the co-authors also, Patricio Munoz, who also presented earlier today. So what we were looking at is understanding the demand side of the blueberry market. And as we all know in this room, per capita consumption has seen a marked increase. So just to show you in this graph here, from per capita consumption was 0.26 pounds per person in 2000 and increased to 277 in 2020. That's a 970% increase. If we would just look at the last decade, that would be like a doubling of the consumption. So along with that, we've also seen an increase in both domestic production and also imports. This is coming from dynamics in increase in acreage in not only in the US, but also in other areas, other regions of the world, also new cultivars being adopted. As you can see here, while we've seen some of the increase in production here in the US, we also see that the rate of increase, or the pace of increase from imports has outpaced that growth in domestic production. So that has generated some of more competitive market. The market, as many of you know, has become more selective, more quality driven. And so there is the importance of understanding what the consumer wants. As we see that increase in the supply of blueberries, we need consumers to purchase those blueberries. So that's what the goal of our studies are, is just to understand what consumers prefer and just what is the value, the economic value that is attributed to those traits, or those quality traits, and that to inform just breeding efforts. So the first paper I'm going to be talking about is willingness to pay for blueberries, sensory attributes for quality traits, and also we looked at consumer characteristics. Because of time, I just may skip that, but if you have any questions, I'll be happy to talk to you about that. So what we were looking at here is to understand how quality traits affect a consumer's preferences for blueberries, how sensory liking, the intensity of those flavors or hedonic attributes, and how that relates to what consumers says they're willing to pay for the blueberries that they have tasted. So for this study, we examined 52 cultivars, so 26 from Florida Southern High Bush, and 26 cultivars in Oregon. So you can see here what cultivars we evaluated each year. So the cultivars from Oregon were predominantly Northern High Bush, although one of them wasn't. But those were evaluated in years 2020, 21, 22. And then, so we collected instrumental chemical analysis, so soluble solid contents, pH, titratable acidity, total sugars, total organic acids, and then we also, we had some measures for firmness. Then we had sensory panels, so those were consumers that were recruited to taste the blueberries. So we conducted the studies for the blueberries that were harvested in Florida. They were tested at the sensory lab at the University of Florida, and the ones that were harvested in Oregon, they were tasted at the Oregon State University Food Innovation Center. So the idea was to have roughly 100 panelists testing each one of the cultivars each year. So we divided the testing in 4 sessions. We had 5 cultivars tested per session, roughly, and so the samples were presented in a randomized order. Then the panelists were asked to first look at the blueberries and state how much they liked the appearance of the blueberries, and then to taste the blueberries and to let us know how much they liked the texture, flavor, and overall liking. And this is just using the standard hedonic scale, 1 equal to the extreme dislike to 9 extremely like. And then they were asked to rate the intensity of sweetness, sourness, firmness, juiciness, and flavor from 0, which is low, to 100, which is high. So after they tasted and evaluated and rated their liking, the sensory liking, and the intensity, they were asked how much would you be willing to pay $3.29 for a 6-ounce clamshell of the sample that you just tasted. So this is what we call a willingness to pay assessment. We use a double-banded dichotomous choice. It's basically 2 questions, and it's a yes or no answer. The reason why we don't ask consumers generally how much you'll pay just as an open-ended question is because sometimes it's like a high burden for consumers, and also they sometimes can give you more unreasonable answers to that question. It's easier when they go to the supermarket, for example, you see the price, you say, I'll buy it or not. So that's the idea of this. Will you pay this? So if they say yes, then there was a second question. So you say yes, there's also a second question with a lower price, with a higher price. And if they say no, they will see a follow-up question with a lower price, just to get an idea of the range of prices they will pay for the particular sample that they have just tested. And our idea here is to be able to correlate the value, the economic value that I assigned to that particular sample to how much they like the sample, and also to the chemistry of that sample. So when we had the willingness to pay data, we estimated different regression models to be able to correlate that data to willingness to pay. The first was with sensory, so we wanted to see, given the sensory value, how much they like a particular sample, how that correlates to their stated willingness to pay. The same with the intensity. So if the intensity scores that they assign to the sample and how that they correlate to the price that they will pay. And then also we did that so this is all subjective, right? Like consumers, it's subjectively how much you like or the intensity that you feel or that you perceive in a particular sample. But we also wanted to correlate that to actual instrumental measurements. So what we wanted to have is a measure of sweetness, sourness, and then firmness. So these are different measures that we included in our regressions to be able to capture what that correlation is. We also, and then we try to control for differences across years, across locations, and also demographic characteristics to be able to tease out the variability due to those factors that are not characteristics here. So here I'm just presenting willingness to pay, so that's the price, as a function of liking score. And here I'm just basically showing, so in the horizontal, the x-axis, the horizontal is the price increments for one additional increase in the score. So for the score for likings is 1 to 9, so one additional increase in the score is associated, for example, with 21 cents increase for flavor. So what this tells us is flavor is by far the most important attribute when it comes to liking. Texture was second, and then appearance was less important in this particular study. So just to give you an idea of what this means, so for the samples that we had, the average liking score was 6.5, and the highest score was 9. So that would mean a difference in 50 cents per clamshell for an average liked blueberry to the blueberries that are most liked by the consumers in this study. Then this is for sweetness, the intensity ratings. Again, sweetness, just by far the most important intensity, and that correlates higher with value and the price that consumers are willing to pay for that. And again, just to give you an idea here, this is a scale of 0 to 100. That's why the numbers are, the increments a little bit smaller. But what that tells you is that basically $1 difference in what consumers are willing to pay for a fruit, or for blueberries that they consider with low sweetness to a fruit that's considered with the highest sweetness in the sample. So now in this graph, from this graph and on, I'm gonna be showing basically what the predicted price will be based on the instrumental values, the quality instrumental values for quality, whether it's sourness, sweetness, and firmness. And this one, so again, here in the horizontal x-axis, I'm going to show the instrumental value or the trait or quality that we're measuring. In this case, it's pH, and that vertical y-axis, I'm going to show the price. So as you can see here, just predicting the price for different values of the pH. And then you'll see the vertical dashed lines. And that just to give you an idea of the distribution of the values that we saw in our sample. So if you see the first dotted line is after 3.20, so that means the 25% of the samples that we examined are below that. The next 25% are between 320 something to 340 and so on. So that will give you an idea of the distribution of the blueberries, pH in the blueberries that we sampled. So here we can see the people so pH is important. We see an increase in the price that consumers are willing to pay as pH increase, right? So that means that some acidity is important. However, we see that starts to decline after a pH of around 3.6. So highest value the consumers assign to blueberries is when it has a pH between 3.4 and 3.8. After that, when you see that pH increasing, then we see a decline in the willingness to pay. Here I'm showing for soluble solid content. Again, we see a positive effect on our willingness to pay as we see an increase in soluble solids. So again, I know that our previous presenters say this is not necessarily correlated to sweetness, but say sweetness there. So as we see that soluble solid content increasing, then we see also an increase in willingness to pay. However, we do see a kind of a plateau here, right? So if you were to think about breeding and trade-offs, there's really not much increment in what consumers are willing to pay going from a content of 15 to 16, right? It's increasing and then kind of reach a plateau where additional sweetness or additional soluble content does not necessarily appeal more, or increment is not there for an additional benefit. For titratable acids, we generally saw a negative effect on willingness to pay. However, we did have a lot of noise here, so it was not as statistically significant at the levels that we like in academia, but we do see that negative effect there. Then we looked at total sugars to total organic, soluble solid content to titratable acid ratio. And again, we do see an increase in willingness to pay as that ratio increases up to a point. Then we see the decline, right, when that acidity gets to increase comparable to the sweetness that consumers are potentially perceiving. So we do see those higher willingness to pay or higher prices in a ratio between 35 to 45. After 45, we do start to see that decline and the willingness to pay. Very similar with total sugars to total organic ratio. We see an increase in willingness to pay with an increase in that ratio up to a point, a 30 ratio. After that, we do again see a decline in that willingness to pay. I'll skip this one. So here we have the total organic acid to total sugar content. I just wanted to show here the relationship between organic, without calculating that ratio, just wanted to show the willingness to pay for various levels. So as you can see here, so the lines increase here means the higher levels of the sugar content, higher willingness to pay. We see a decline as organic acid increases. And what we can see here, so from 140 milligrams per grams, or 14% dry weight concentration in organic acids, we just see that regardless of the sugar content, we see very similar willingness to pay there. That potentially means that a higher acidity levels, even when you have the higher levels of sugar concentration, we still see that it's not necessarily enough to offset that increase in organic acid and decline in prices. So fruit for size, we also saw an increase in the willingness to pay with size. I have to mention here that we did not see a statistical difference in the price between 14 millimeters and 17 millimeters. After 17 millimeters, we found an increase in a willingness to pay for berries. And that's something that has been mentioned, consumers wanting bigger fruit, right, or larger fruit in the market. And here I'll have firmness. I'm going to skip this. But generally speaking, what we all know, there's some firmness that is demanded in the fruit, but for very hard fruit, the willingness to pay decline, right? Like if you had too much fruit. So there's a sweet spot there where consumers are looking for that crisp fruit. And then just generally, we did ask the consumers in this panel, what were the traits that they care more about? And freshness was mentioned as the very, very extremely important by 92% of the respondents, followed by flavor, which is very interesting. Again, like freshness, we saw freshness coming up a lot in the surveys. Okay. Then we had consumers that consume blueberries, but very infrequently. And so we asked what are the reasons for their infrequent consumption. So price was cited by 30% of them, and short shelf life by 24% of them. I'm going to talk real quick. I'm running out of time, but I'm going to talk real quick about another survey that was more representative, and we see these topics coming up a lot. Just going to briefly describe another study we did looking at quality-related descriptors in the labeling or packaging of blueberries. So what we did is an online survey using a more representative panel of U.S. consumers, and we have 3,200 participants. And what we did is to use a basket-based choice experiment. So when you go shopping, you see a basket and you select the products that you want. We presented them with 14 fruits. One of them was blueberries. And what we wanted to taste is quality descriptors. So, like jumbo, when you see the blueberries jumbo, that's something that you can actually see. These will be descriptors that you don't necessarily are able to observe, but you have to taste it, right? So we wanted to convey information about crispiness, sweetness, a longer shelf life. So the words we used was crunchy, sweetie, stay fresh. And then we divided the groups. There were very similar groups, so we can make comparison across those groups. One of them saw blueberries in the basket, just the blueberries with no labels. Then another group, the second group, saw crunchy. The other say stay fresh, and the other group saw sweetie. And then they evaluated different baskets, and we varied the price points for the different fruits. Okay, so just to summarize what we found. So we found that the stay fresh indicator of shelf life, was linked to an increased likelihood of portions in the blueberries. So imagine you go and you see, you go to the store or you see online. So how likely are you to actually select the blueberries? So if they had to stay fresh, those blueberries were more likely to be chosen or bought. The terms emphasizing texture, like the crunchy or taste, sweetie, were actually not different from not label. So they did not increase the likelihood of purchasing. Now, when it comes to The labels Stay Fresh, again, and Sweetie, they were also linked to reduced sensitivity to price changes compared to unlabeled blueberries. So what I mean by this is that if you go to the store, you see higher prices, you're less likely to buy that product, right? So as prices increases, consumers are less likely to buy a product. So how sensitive you are to those price changes. So the Stay Fresh or Sweetie reduce that sensitivity or that decreased likelihood that you will purchase a product when you see higher prices compared to no labels. We also found that all of the berries were likely bought together instead of like competing with each other. They're generally complementing each other when consumers are looking buying a basket. Then, just to finalize here, just very important, we also asked consumers here the reasons for infrequent blueberry consumption. Price, 55% of them said there was price. Short shelf life, 22%. So very similar to what we saw in the responses with the sensory panel, with price, again, just the main motive or barrier to blueberry consumption, following by shelf life. So these are very consistent results. The attributes they care more about, freshness. So something else to consider as we think about what consumers are demanding in the market. So with that, running out of time. Thank you so much. If you have any questions, I'll be around, so please let me know.
Thank you so much for your time and expertise, Dr. Canales. Our last but not least speaker is Dr. Jonathan Oliver. He's an assistant professor at the University of Georgia, and today he's going to talk about organic blueberries disease management.
So thank you. Yes, I am Jonathan Oliver. I'm the fruit pathologist and extension specialist at the University of Georgia, based in Tifton. And today I'm going to go through the results of several recent trials that we've done looking at management of diseases in organic blueberries. So organic blueberry production in Georgia ranks 4th in the country in terms of acreage, with over 1,100 acres of organic blueberries. But this is fairly small relative to our total production, which ranks 1st in terms of acreage in the country. And this is largely due to the fact that the warm and wet climate in Georgia makes organic disease management pretty challenging, and there are relatively few organic fungicides with proven efficacy for blueberries. The active ingredients that are currently used for organic blueberry disease management in Georgia include various oils, sulfur products, copper, and biofungicides. So biofungicides are materials that include either microbial organisms or extracts from the growth of those microbes. So in recent years, there's been a lot of interest in developing new organic fungicides. And so there are some based on essential oils and also some other newer biofungicides that have been developed. And often these products show promise in the lab and in other cropping systems or other cropping situations, but not always in the field. And so we really wanted to look and see if we could add some information about the efficacy of these products in the field for the management of the primary fungal diseases of concern in blueberry production. Some of these products that have been developed in recent years and are labeled for use in blueberry production include TimeGuard, Timurex, and Cinerate, which include as an active ingredient thyme oil, tea tree oil, or cinnamon oil, respectively, as well as Howler and Thea, which include a couple of bacterial strains, and also which includes polyoxin D-zinc salt. So some of those diseases of concern in blueberry production in the southeast vary across the season at what's most important, but typically these diseases or types of diseases mentioned here are the ones of concern. These stretch across the season from the late dormant period through after harvest. See that early in the season we have issues with Exobasidium and mummyberry, and that moves into bloom through harvest. Fruit rots become the primary disease concern, and then leaf spots are problematic from prior to harvest all the way through the after harvest period in Georgia. And so some of the trial work I'm going to show today focused on really that early, one of the early diseases in the season, mummyberry, fruit rots, which again are important from bloom through harvest, and the leaf spot diseases. In addition to these seasonal disease issues, other problems that affect blueberry production in Georgia include blueberry replant disease and root rot. So ring nematodes have been associated with poor plant growth, high plant mortality, and premature decline of plants. And this is especially true in sites where southern highbush blueberries have been planted in a field that already was in production with either rabbit-eye or highbush previously. And so there are these replant sites. And you can see some of that poor growth in those pictures on the right. Ring nematodes are likely not the only thing involved in this problem. It's believed that Phytophthora root rot may also be responsible for the decline of young plants. Basically, it's kind of a 1-2 punch for the roots of these plants being affected by both nematodes and Phytophthora. So in managing replant can be challenging, but for today, I'm going to go through very quickly the results of several different kinds of trials that we have done, looking first at anaerobic soil disinfestation. So what is anaerobic soil disinfestation? For one, it's a mouthful, so I'm going to be saying ASD when I refer to it over and over, but it is an alternative to fumigation. And soil fumigation is really the standard management practice used in blueberries to reduce disease and nematode issues in these replant sites. And so anaerobic soil disinfestation is an alternative to fumigation that has proven effective for reducing soil-borne diseases in other fruit and vegetable crops, but ASD has not been utilized previously in blueberry production. So the way ASD works is soils are amended with certain carbon sources, they're covered in plastic tarps, and they're irrigated to field capacity to remove basically the oxygen and create anaerobic conditions under those tarps. And then in those anaerobic conditions, those microbes that are present and are favored by those conditions will reproduce rapidly as a result of the carbon sources that were added, and they will generate byproducts that are toxic to soil-borne pathogens. So in tomatoes and strawberries, ASD has been used fairly effectively, and it's becoming more popular and a lot of research is being done into ASD in places like the Netherlands and Japan as well. But we wanted to look at some of these different carbon sources and their use in blueberry systems. So the preferred carbon sources that are used in ASD typically include substrates with a lower carbon to nitrogen ratio that can basically allow for the rapid growth of these microbes. And so brewer's spent grain at 15-to-1 carbon to nitrogen ratio is an example of a common carbon source used in some of these other crops. However, blueberries in the southeast are typically grown in soils amended with pine bark. Pine bark has a much higher carbon to nitrogen ratio, and also it's much harder for pine bark to break down. It doesn't break down as quickly, so it's not as available for these microbes. So it's unclear what impact using pine bark would have in this system. So to investigate the impacts of these different carbon sources on soil-borne diseases and blueberry growth, we did a field study that we started in February of 2022 to compare fumigation, kind of the standard practice, to anaerobic soil disinfestation with 3 different carbon sources at the Alapaha UGA blueberry farm. So our field trial in brief was set up with basically we did plots of 75 row feet approximately is what we evaluated. 25 plants on 3-foot spacing were established in each plot, and this area of the field had a history of problems with Phytophthora and ring nematodes. 5 treatments were compared in a randomized complete block design with 5 replicates. And these treatments were pine bark covered with plastic mulch. That was our control. Pine bark and fumigation was another type of control. And then we compared 3 different ways of doing ASD. One that just relied on pine bark, one that involved pine bark and cottonseed meal. Cottonseed meal is a potential nitrogen source that's used in organic blueberry production, and so we wanted to increase the amount of nitrogen present in these soils to see if that would help stimulate the process. And also we did ASD using brewer's spent grain. So what we did is we bedded, incorporated these carbon sources into the beds, and we bedded it up, we covered them in plastic, and we irrigated the ASD treatments to field capacity. And then we left these plots for about 4 weeks, at which time we began to rip the plastic coverings. And then at about 6 weeks or so after the initial irrigation, we began to plant back into those plots. And we used Southern Highbush cultivar Optimus that we planted in April of 2022. And we looked at nematode counts, Phytophthora incidence, and plant growth and yield during 2022, 2023, and 2024. So what did we see? Well, the before and after of the nematode counts indicated that fumigation, as well as all the ASD treatments, significantly reduced the counts of ring nematodes present in these plots, and that was not true of the control treatment. And for Phytophthora, we saw that while the brewer's spent grain ASD treatment was comparable to fumigation in terms of root rot, the pine bark ASD and the cottonseed meal ASD actually had more Phytophthora root rot. And so something we observed, and you can see here at the bottom, was the root zone stayed very wet for months under this plastic. And that makes sense because we'd fully saturated it. It's covered in plastic. We just basically ripped holes where we planted the plants into. And so it was potentially great conditions for Phytophthora, which loves these wet conditions. But we didn't still see an increase in Phytophthora in the brewer's spent grain, indicating that maybe it did have some efficacy against at preventing issues with root rot. But unfortunately, the plants grown in the brewer's spent grain amended plots showed lower plant growth, fruit yield, and fruit size relative to all of our other treatments. So you can see that pretty clearly in the first graph, plant heights. There's a significant reduction in the height of these plants, which of course were all the same size. They were planted from 1 gallon pots originally. And then, so they grew poorly. They had relatively lower yields. You can see those green bars on the right where we show the average yield per plot in 2023 and 2024. And the fruit from these brewer spent grain plants was weighed less and was relatively smaller. So even though it may have had some benefits for disease control, it was unfortunately not an appropriate soil amendment for growth of blueberries based on these parameters. So all the ASD treatments reduced the counts of ring nematodes comparable to fumigation, but the other results were fairly mixed here. The pine bark and pine bark plus cottonseed meal treatment had relatively higher amounts of Phytophthora root rot than the brewer spent grain treatment, the fumigation treatment, or the control. Plant growth, fruit yield, fruit size were negatively impacted by the use of brewer spent grain as an ASD carbon source. So after this work, we're really left with the important point that it's important to identify an appropriate carbon source for ASD that would work in blueberries. You have to balance both things. You want a carbon source that will allow you to conduct the disinfestation correctly and reduce your soil-borne diseases, but you don't want something that's ultimately going to negatively impact crop growth, because that's kind of the whole point, right? So that was an unfortunate finding, but now we know about these carbon sources and their use in ASD. So I'm going to move on and go really quickly through a bunch of fungicide trials we did looking at mummyberry, fruit rots, and leaf spots. So mummyberry, as you probably know, is an important disease impacting blueberry production all over the place. It's more of a problem in South Georgia on rabbit eye blueberries because the life cycle of the mummyberry fungus doesn't quite sync up real well with the southern highbush. But southern highbush are certainly susceptible and can get infected elsewhere. This disease causes this shoot blight, which you can see here in the second picture. It also, ultimately, the fungus infects the fruit and it shrivels the fruit from the inside out, leaving behind these dried husks. You can see on the far right, thus the name mummyberry. And these dried mummies fall to the ground. And from those mummies in the spring emerge these cup mushrooms, which you can see on the left side of this slide. And these cup mushrooms release spores that start the cycle all over again. If you can remove those mummies from the field by either raking them out or burying them over an inch deep, you can have some level of control of mummyberry disease, but this is generally just not sufficient for good control. As you can imagine, especially in some of these organic plantings, weed control is already a challenge. It can be hard to find those mummies. They hide in the grass and hard to get them out of there. And if you don't bury them deep enough, that cup mushroom can form right through the soil. So it's really limited. And mummyberry, I would say, for organic production in Georgia is really one of the big limiting factors over time. Initially, new planting may not have issues, but mummyberry builds up. And so this is really the disease that often forces organic blueberry growers in Georgia to break down and say, okay, well, I've got to spray something and control this. And they have to spray a conventional product to control it and lose their organic status potentially. But otherwise, it more or less takes it out of organic production. So chemical controls are very effective. The conventional controls, things like Pristine, Luna Tranquility, and the DMIs are very good or excellent if applied from green tip through bloom. For organic production, the list is much smaller, and the efficacy is limited for organic production. So we wanted to look at some different organic products and conduct some different trials. So we did trial work in Baxley, Blairsville, and Toccoa. We did randomized complete block designs with 5 replicates. The treated plots generally were 1 or 2 plants, depending on the size of the plant and how much fruit we could assess off of the plant. The fungicide applications were made every 7 to 10 days, so very frequently, from green tip all the way through bloom, and that's at the recommendation of the different organic fungicide companies and labels to apply them on a frequent basis. And we made applications using a backpack, and we assessed disease by either counting the number of strikes, those leaves that shrivel up, or by determining the incidence of mummified fruit among the fruit we harvested. So in 2021 in Blairsville, our results look like this, and I've color-coded everything from here on out. If it's in green, it indicates a significant reduction in disease. If it's in yellow, it indicates a result that was not significantly different versus the untreated control. And all of the organic fungicides in these tables are going to be bolded, and the italics is either for the untreated control or a conventional fungicide, which in some cases was used as a comparison. So you can see that in this trial, in terms of shoot strikes per bush, at least on a Clockney where we had enough disease to evaluate, we did see a reduction in shoot strikes per bush by using several of these fungicides. Unfortunately, while some organic products reduce disease in this trial, they only reduce disease sometimes, and a lot of our other subsequent trials, we've not seen as much efficacy. You see that trial on the right, there is some green there, but that's actually all from the conventional fungicides, which were tested alongside that DART, the organic fungicide in that trial. And in some cases, with trial work, we have situations where we don't have enough disease to evaluate, and in other cases, we have plenty of disease, but we just did not see efficacy, as you can see in the bottom right on the Brightwell. So moving on to fruit rot trial results. There are 3 major fruit rots that impact blueberry production. You have anthracnose rot, alternaria rot, and botrytis gray mold, and also causes blossom blight. And typically with these fruit rots, infections begin around bloom, and they remain latent until the fruit ripens. So the fruit can get infected from its very earliest stages back at bloom, and then the fungal organisms that cause these rots will just hang out there until the sugar levels begin to rise, and then the organism will kind of wake up and begin to shrivel these fruit and form spores, and et cetera, that you can see on the right. Warm weather during bloom or harvest favors anthracnose. Cool weather favors alternaria. Botrytis is particularly problematic when we have wet weather near bloom or if we have freeze-damaged tissues present, and then it can cause a blossom blight as well. Control of these in conventional blueberry production typically involves fungicide applications from bloom through harvest. So for our trials, we applied organic fungicides from bloom through harvest every 7 to 10 days, so that resulted in about 7 or 8 sprays in most of our trials. Treated plots range from 1 to 4 plants, depending on what the size of the plants were and how many plants we could harvest the fruit from, because we wanted to harvest at least 200 fruit from these plants that we could evaluate for fruit rot incidence, and so we evaluated rot incidence based on visual signs. So in one of our trials from 2021 from Baxley, we saw some efficacy from Time Guard, Timurex, Double Nickel, and Howler, and so we were interested in following up on that and conducting additional trials. And I'm very sad to say that in all subsequent trials, we did not see any efficacy of even some of these same products, but any of the other products we tested either, against these fruit rots. And in fact, in a couple of cases, we saw some phytotoxicity. Pure Spray Green caused this darkening of the calyx. You can see in that top image on the right that, of course, would make the fruit more difficult to market. And Time Guard, in at least one of the years in 2024, in both rabbit eye and southern high bush, both here and in trials that our collaborator in Florida did, showed this significant phytotoxicity on the fruit. You can see on the bottom picture there, that actually led to more rot in these fruit. And so that was a disappointing bunch of follow-up trials. And moving on to leaf spots, we have 2 major leaf spot diseases in Georgia on blueberry. Septoria leaf spot here at the left, and blueberry leaf rust shown at the right. And leaf spots impact blueberry production because they ultimately limit photosynthesis, and this will cause the plants potentially to defoliate early if the leaf spots get bad enough, and it will reduce return bloom for next year, so it will impact yield next year. Applications to control leaf spots are typically made from early cover sprays, so prior to harvest, after bloom, through really the end of the season in Georgia, which in most cases is in October, where we anticipate the plants starting to enter dormancy in November. So for our leaf spot organic trials, they were conducted in a similar fashion, except in this case we sprayed every 7 to 10 days in some of our trials earlier in the season, knowing that these leaf spots can develop in the spring, and in some trials we sprayed plants later in the season to look at kind of the development of leaf spots after hedging events that occurred during the summer. And the applications were made with a backpack again, and we assessed spot incidence and severity being the number of spots per leaf or the area of the leaf estimated to be affected by spots. So we did see some significant results from Pure Spray Green in these trials. It did reduce leaf rust in terms of incidence and severity in a statistically significant way relative to the untreated control. And you can see that in those top 2 trials. And then here in this bottom trial, Howler Evo reduced leaf rust incidence and severity as well. So in the bottom trial, we included several, this was done at the research farm, so we were able to include conventional fungicides in this trial. We saw the conventional fungicides listed at the bottom did extremely well at controlling rust, and Howler was kind of in the middle between the very good conventional products and some of the organic products that were less effective. But it did have a significant reduction in terms of the incidence of leaf rust, maximum severity, and the leaf area affected in this trial. And so Howler Evo is another formulation of Howler. I had mentioned Howler in a previous slide. It's kind of the newer formulation of Howler, but it has the same active ingredient. But unfortunately, most products did not show efficacy versus leaf spots. So we have these multiple other trials where we didn't get any efficacy out of any of our tested products. And in fact, in that same trial where there was fruit damage caused by time guard, there was also leaf damage caused by time guard as well. So a lot of negative results that I showed. I'm sure that was thrilling. But let me go to the organic summary, because I think it really is important to kind of put it all together here at the end. So organic fungicides have been included in 21 trials in total since 2020. And those trial results are shown at the right. You can see the list of fungicides and the 3 different types of diseases we looked at: mummyberry, the fruit rots, or the leaf spots. And it's shaded in green. The cell is shaded in green if that particular fungicide had efficacy in at least one trial for each of those different diseases. And you can see that overall, we had a lot of trials that did not produce any efficacy, but we did have 3 products, Howler or Howler Evo, also, and Pure Spray Green, which did show efficacy in more than one trial. And so I've been asked, why we didn't see more efficacy. These results to me indicate that the efficacy of these products may be limited, and it may be limited by different conditions, or in situations of high disease pressure, the efficacy of these fungicides may just be overwhelmed. Again, we do have high disease pressure in most cases in Georgia due to our wet and warm conditions. And so that's a summary of those. And in conclusion, overall, the ASD work definitely indicated the importance of identifying a carbon source that effectively reduces soil-borne diseases without negatively impacting subsequent blueberry growth. Overall, in our recent fungicide trials, most of the organic fungicides did not demonstrate consistent efficacy. However, Howler, also, and Pure Spray Green may provide control of certain diseases in at least some situations and conditions. And certainly, I think you'd probably agree that we need additional organic options if we're going to manage these major blueberry diseases effectively in Georgia. And with that, I'd like to thank you for your attention, and also thank the funding we received from USDA for some of this organic work with the essential oil fungicides, as well as various funding sources that funded individual trial work over the past few years. And with that, if you have any questions, by all means, I'd be happy to answer it. Yep. Well, some of the trials, certainly nothing did just as well as spraying anything, unfortunately. But as far as what has demonstrated some efficacy, so a rotation of Osso and Howler may be an option. Some of these organic fungicides are not really likely to lead to resistance development, so if you didn't want to do a rotation, you just want to spray the same thing over and over, that may be okay as well. But they may have slightly different profiles as far as what pathogens they're, because sometimes we just assess rots overall, so there may be some distinctions to be made between their efficacy against certain rot pathogens and certain other rot pathogens, and so a rotation may allow you to cover your bases there a little bit, and so that may be a good idea. Mm-hmm. On the fruit or the blossoms? Yeah. So also is believed to definitely have efficacy against botrytis. So there's a comparable product, actually has the same active ingredient, as also called PHD, which is recommended in blackberry production and other systems for use against botrytis. So also I would expect to have some efficacy against botrytis. Botrytis is really a hit or miss issue for us on blueberry. We see it it can cause major issues on the blooms, and in certain years, yeah, it'll definitely cause issues on the fruit, but we don't always see it. So for a given trial, we didn't necessarily see botrytis specifically. Anything else? All right, thank you.
Thank you so much, Dr. Oliver, for your time and expertise. With that, that's all that we have for today. Thank you, everybody, for your time and attend all the presentations. See you tomorrow. Thank you.